And so here we are, ready for Dr. Dave Esslinger. And before I let Dave get started, I do want to say thank you and um, a fond farewell to our friend Dave Esslinger. For those of you who do not know, Dave and his wife Sandy are going to be moving out of the area at the end of this month. So I don't know what Dave's plans are, but this might be the last time we have this presentation. So definitely worth the recording and definitely worth us keeping it for posterity. But my thanks and gratitude go out to Dave, not only as a mentor, but as a friend. I probably wouldn't be president without his help. So um, his help has definitely made life a lot easier for us all, I'm sure. So, if you haven't signed up for his session on Saturday morning, I don't know how many spaces there are left. There were a few, four or five, I think, last time I looked. But if you need a good refresher, a hands-on refresher with the microscopes and see what we see in the pond and other waters of Corkle, this is going to be a great opportunity for us all. So, thanks again, Dave, for all your hard work as president and associations with us as an association. And, hopefully we'll have an opportunity to say farewell officially. But I know time there is of the essence. So I just wanted to say this thank you publicly for all. And hopefully everybody at the end will be able to join in too and wish you well. Hopefully we'll get to see you before you leave, other than Saturday. But with no more words from me, or just the one or two, we're here to see Dave. I'll let him do his introductions and his background. Obviously, he's the man with the experience. Uh, and we've had his presentation before if we've gone through our Master Naturalist program, and he's been one of our instructors for us. This should be a refresher, but it depends on when you went through the program and whether Dave was teaching that session. But anyway, turn it over to you, Dave, and thanks again.
All right. Well, thank you, Colin, for those very gracious words. It is, as I mentioned to Michael, it's a pretty bittersweet thing to be leaving my group of friends here in a place I've lived for, well, since 1998. So not quite 30 years, but it's exciting to go out and try something new. There will be at least one South Carolina master naturalist out there, Bev Gulis, who is a good friend of Betty and Popilo, who is on here as well, and also has become a friend of mine out there, lives in the same subdivision that I'm in. So that was a fun happenstance. So I already have a master naturalist buddy out there. But by way of background, most of you probably know I'm an oceanographer. Worked for NOAA for 23 years, and I came down to South Carolina from Fairbanks, Alaska. But I really got interested in microscopy when I was 12 years old, and I traded an erector set. For those of you who remember, that's a little bunch of metal pieces and nuts and bolts that you screw things together and build. And I had one that I was tired of playing with, and my friend had a microscope he was tired of playing with. So we swapped. And I've been fascinated ever since. So tonight I'm really trying to give you an introduction to what I'm hoping we will see out at CACA, and also to, talk about these, excuse me, alien worlds. And I really think that's a good description, and they really are all around us. If this is going to work for me here. Whoop. So to start off, a few things to ponder on here. What do I mean by alien? Not in the extraterrestrial sense, but in the sense that these organisms are bizarre. They are living in a world that is completely different than us. It is almost as if they are on a different planet because the laws of physics, they're still the same laws, but they behave very, very differently at this really tiny scale. And in adapting to that, they have come up with these crazy lifestyles and life cycles that are very different than what we're used to. Being alive and dead are seem not to be the definitive states that they are to us, because things, you would swear they were dead and they just pop back to life when you put a drop of water on them. So, and they can do that multiple times. So they're not really dead, but they are in resting states where they can remain for decades, and then just pop right back out and be reproducing within a few minutes of being hydrated. And then the whole way that we usually break down the world into plants versus animals, that is definitely not a clear demarcation. And as I mentioned, why are these worlds alien? I'm gonna tell you just a little bit about the physics of the environment, and then we're gonna try to look at some examples of how they move to and adapt to that. Talk a little bit about some of their reproductive strategies and their feeding strategies. I don't have any really good slides on spores and stasis, but those are some of the things that they do. We as humans, we can't really say, hey, it's hot out today, or I don't like the current situation. I'm going to go into a resting state for a decade, and maybe things will be better at the end of that. And you won't age during that time. Might be nice to be able to do that, but we can't. But these little guys can. And another thing I want you just to appreciate is, where are these aliens? They are all around us, because the worlds they live in are small, like your bird bath. Many of the pictures that I'm going to show today came from organisms collected out of the 3 rice tubs that are right outside of Kaka. So that's one of my favorite places to collect things, because each of those are different, even though they are sitting right next to each other. So within a couple of feet apart, each of those tubs have a different environment. They are at the micro level, they are different worlds. And these environments are everywhere. The first tardigrade that I found was living on the underside of a magnolia leaf, because you get a little bit of water there. And so just the condensation and humidity, that level of humidity, was enough to support microbes and things that feed on the microbes. All right, so here's a little hard science, very, very little. There is this thing called the Reynolds number, and it's a way of looking at the world and comparing how viscous is your environment compared to how big you are and how fast you can move. And that essentially says, how sticky is the environment that we move through. If you're at a high Reynolds number, you don't care that you're walking around in through the medium you are. If you're at a low Reynolds number, you might. So for humans, when we're walking around in air, the Reynolds number is about 10 to the 7th. That's 10 million times, right? This is scientific notation. I think we've all seen it. And you've probably noticed when you get in water, water is 1,000 times denser than air. And so that makes the Reynolds number drops by a factor of 1,000. So it's about 10 to the 4th. And I hope you've probably noticed it's harder to walk through water. You've really got to exert some effort. And as I've recently done, I have a pool here in my house, and my grandson is trying to swim to me. He's just learning to swim, so I'm constantly backing up to make him swim more, right? So he becomes a more capable swimmer. It's not just being mean to him. But what I notice is, some of the time, if he's close enough, he doesn't even have to swim, because he just gets caught in my wake, and I'm pulling him along even when I'm 2 feet away. That would never happen if we were in air. It's happening because we are in a denser, more viscous medium, which is water. That's what happens to us that we've experienced when we go make that change of 1,000 times. Now, for the protozoans and the things we're going to look at, because they are so small, their Reynolds number is 10 to the minus fourth. So that's 8 orders of magnitude. That's 100 million times thicker environment than we are in when we're in water. All right? Think about that. They're living in jelly. They are living, literally, it's like they live in jello. So what that means is that some of the laws of physics that we think about all the time don't really matter. Like gravity. They will barely sink. They will sink, but it's very slow. Viscosity or inertia is just disappears because the viscosity dominates everything. So if they're not actively trying to push their way through this thick world, they stay still. It's really fascinating. And at the other end of that, at the high Reynolds number, so I just learned this summer when I was teaching camp. There was a guy up there. He's studying shark skin because sharks live at very high Reynolds number. They're up like us, but their skin and their scales are adapted to reduce the viscosity or to reduce drag, so that they're at the opposite end of this range. They live in almost a frictionless environment, which is why when you see them gliding along, they barely move because they're at a super high Reynolds number, and the little structure of their scales reduces the drag coefficient on them a great amount. Really cool stuff. That physics tells you about our natural world. All right, so if you want to move, okay, so that was all the boring science, most of the boring science. And then from here on out, I'm going to just try to show videos and talk about them. So I hope you're appreciative of this kind of show and tell approach. And it might be a little bit more scattered than what I generally like, but as I was looking through my microscopy stuff, I have over 600 videos that I've recorded. And so trying to pick some, I told my wife that. She just looks at me in all serious. She goes, "That's too many." I'm like, "Yes." Thank you, dear. Oh, I'm not going to do that many. But when you are, did I skip something? Yep, I skipped something. Okay, so when you live in this jello environment, it turns out if you do the physics of, if you have a good firm oar, like we use when we row a boat, you can't use that. You just stay in place. So because and that makes sense if you think about it, because you push forward and then you push back, right? So microorganisms have figured out ways to have bendable oars, and some of those are cilia. And so this is a little cilia. And hey, Colin, can you guys see my cursor on the screen? Okay. So if you look, I know it's a little a little hard to see as I go through this, and I'm focusing in and out to try to show you that there are cilia right around the edges of this. And let's see. So those are cilia. That's the cilia at a zoomed in pretty good. Here's how efficient these little suckers are. And this is the leaf that you see over here. These are the individual cells in a duckweed. All right, so duckweed is really good. We're gonna try to grab some duckweed and look at it, and I hope you'll get some clues too on how to look at things. But these are individual little paramecium-like cilia, so they have little tiny bendable hairs that are waving in motion and together so that they can swim. And then another way, a type of a bendable oar, is to have multi-jointed appendages. And this is a cool organism. This is called an ostracod. So this isn't a this isn't a protozoan, this is a metazoan, meaning multicellular animals. We are metazoans. Protozoan, only one cell. So this ostracod is, if you can imagine, a shrimp, but instead of having its shell being flat across the back, it's grown around it almost like a clamshell. And so this ostracod, you can see it moving around, and it's able to manipulate its environment with its these little appendages, legs, I guess we can talk them. And then another one here is, this is a calanoid copepod. We'll see right here, and this is not the best image, but you can watch it. It zips, and in this particular slide, I got to try to follow it. Notice how it moves and then it instantly stops, because it's living in jello. So there's no coasting, and it would be the same if there weren't all this detritus on here. Come on, there you go. And so he's just tootling around, but using these bendable oars that are jointed appendages. All right, one of the other things that works really well to move something around through jello is a corkscrew. And flagellates, which is just anything that has a flagella, use something that looks very much like, or that works like that. This particular flagellates, and lots of flagellates have multiple flagella. Really, is that all? I cut that a little too short. So it has actually a flagellum out in front that is moving along there. You could see it a little bit. You can see it's actually going through the water and pulling it along. And it's got a second flagellum that just is dragging along behind it, serving no purpose as far as I know. So flagella and corkscrews. We hopefully will see some, I don't think I put any videos in them, but a lot of bacteria and spirochetes have this spiral form too. And so they just are able to just move and corkscrew their way through the water. Because again, think of it as jello, or even something even thicker. Whoops, we've seen that guy enough. All right, here's one of my favorites. Gliding along a substrate is another way of doing that. And believe it or not, these are diatoms. This is the way that diatoms, we think about them as, oh, they're phytoplankton, they're just floating around. But many of them, like this one here, this is Basilaria paradoxa. And it got that name. It was the very first diatom to be named or identified. And so that's all of the diatoms are in the group Basilaria phyces. So they are named after this particular organism. And but the guy didn't know whether it was an animal or a plant, because that was the way you thought about things at that time, because it was moving like this. And so he called it was a paradox for how it was moving. And if we pause it here, notice what it's doing is each of these is one cell. It's a single-celled animal or organism, excuse me, single-celled organism. And diatoms, remember, have a silica shell that's composed of two halves, and it fits together kind of like a lid and a top lid and a bottom lid. And the along the midline of that of each lid is this slotted groove for many diatoms called a raphe. And they exude some of their intracellular content, so their cytoplasm out of that the little slits in that and slide and use that to glide along. And then they pick it back up because they don't leave little trails. But this particular diatom gets around by the whole kind of colony of them. So these are all sister cells because when they divide, they don't separate. They just slide next to each other. So these are all clones. And then here's some other diatoms you can see moving around. There's one up here. There's another one up here. This is not a diatom. There's another one right down here. Most of these are Nitzschia species, or they're from the genus Nitzschia of these pinnate diatoms. So really cool way to move. And then, of course, the famous way of the famous gliders by, oh, don't do, there we go, thank you, of doing their, of moving are amoeba. And so this is, what I'm doing is I'm focusing up and down. So we're looking at different heights in this amoeba, and it has this three-dimensional structure. Man, I asked PowerPoint to compress my videos so they weren't quite so big, because this was a 30 terabyte thing when I started. So it doesn't look that great. I'm sorry about that. But look at this amoeba. It started on, I think, the cover slip of a slide, and it's extending out and down, looking for a good substrate to crawl on. And then this is one of the amoeba where it puts out different pseudopods to test which way to go. And you can see here as I'm still focusing up, it has a three-dimensional structure in it. So it's searching in all directions and going in whichever direction it is. I don't know why it decides to go in one of those. All right, because these organisms, this is one cell. It doesn't have a brain. That's one of the things that is just so mind-boggling to me, is these little organisms do everything we do with one cell. The protozoans, we will see, as we did already, some of the metazoans, some of those have brains. Here we are again, so it's reaching up and down. And they just reach out, trying to find things. Very cool. Amoeboid motion is usually pretty slow, so it takes a little bit. Here is another amoeba and a diatom up here. Notice how this amoeba doesn't have all those crazy pseudopods that we think about. This is a different class of amoeba, and it just has this thin hyaline edge up here where nothing is in it. It's got a cytoplasm in here, and you can see the individual components in there. It's got some vacuoles back here where it's ingested different foods. And it will eventually, it just kind of does the equivalent of pooping them out there. It excretes them by just leaving a lump, some of these together or behind. And then I believe the next one it should be, oh no, I had one that was sped up 10 times faster, but you got it. Or you understand, amoebas glide, right? They're pretty cool. All right, so let's talk about, so that's some of the ways that these organisms can move in this jelly-like environment. Some of the other ways in which they are really, really bizarre is their life cycle. This is a proposed life cycle for an organism called Pfiesteria. Do you guys remember maybe 20 years ago, there was what was referred to as the Pfiesteria hysteria, because Pfiesteria is a toxic dinoflagellate that was causing some very large fish kills, especially up in North Carolina. It's interesting in that it is it's not toxic to us, but it's toxic to fish, and it can be toxic to them at very low levels. But people were trying to figure out its life cycle and where it came from, and why all of a sudden it was popping up as a harmful algal, and this is what they proposed as its life cycle. So, you know, we are familiar most of the time with something like a complex organism. If we think about other, humans, we have a gestation, fertilization, gestation, and then a little baby is born and it grows up to, and it's basically a baby's a little version of the adult. So lots of organisms go through that, and then some of them have incomplete metamorphosis, right? That's the insect equivalent of what we had with like grasshoppers and things, where they have different instars and they just molt and mature to adulthood. And then the complicated life cycles are like the complete metamorphosis, like butterflies have. So that there is a egg, a larval, a pupal stage where they're in a cocoon or a chrysalis, and then the adult. So 4 stages. Okay, in our regular world, we think about that. That's quite a few. So these amoeba, or this one organism, has been hypothesized to have 18 to 20 different stages like that. And about a third of these are toxic, and none of the rest of them are. So it really is, it's a hyper extreme example. That's probably not even a real word, but of how complicated things could be, where an organism has both, this is a dinoflagellate, so it has flagella, and it can actively swim around a little bit, but it also has a benthic amoeba stage, and then it goes into these resting cysts. It has sexual reproduction with these different gametes, and mostly it has asexual reproduction. Pretty crazy. However, for most organisms, when we think about reproduction of protozoans, so one-celled animal, binary fission is what most of them go through, and they essentially, one cell divides to become 2 cells. And yeah, this is a little video. Let's see. So here's a couple of paramecium that are just at the final stages of dividing. I usually they manage to get away from me before we see them split, but you can see again, these are ciliates, so they've got those little bendable oars on them moving around. They've got internal parts in here. This is called the contractile vacuole that's moving, which they have. They just kind of have parts of their body they fill up with water and squeeze it out to excrete their water, but they are slowly getting ready to break apart over here. That's all I have for them. This is those diatoms, and these are Nitzschia species again, and that is remember how I said they were made out of these 2 different shells? They're called tests, or valves, but each valve, as they split apart, the top valve grows an inside valve, and the bottom valve also grows an inside valve. And then you end up with one cell that is slightly smaller, 2 daughter cells that is slightly smaller than the original cell. And that is an interesting thing because it means that one can almost think of this initial cell as, and people have said, oh, it's like an immortal cell, because they don't actually die of old age, they just divide. And the top one never gets smaller. So this is a schematic of the side view of, okay, centric diatom or round diatom. You've probably all seen pictures of those, right? So that, and they say, oh, it looks like a pillbox, except most of us don't get our pills in boxes anymore. So it's not a good analogy anymore. But these pillbox diatoms, if you will, so that the lid is going to come off and it makes a new bottom piece inside it out of silica. So it becomes a new cell. And the bottom piece makes a new inner piece. So it's been flipped over here. And so that green is the new inside of this blue. And so it's a slightly smaller cell. And you can see as one continues through that, you get, there's always these really big cells. Then there's another size, and there's even smaller ones. And they're all the same species, but they get smaller and smaller until at some point, there's just not enough cellular material in there to be a viable cell. And that actually works out pretty good, because that's the point. All through this, there's been no genetic exchange, right? And we know in our normal non-alien world, you need to have genetic exchange so that organisms can evolve, can adapt to new environments. They will be a little more stable and resilient to change. So without that, all of these diatoms have exactly the same genetic material. They are essentially clones of each other, but different size. At some point, when the cell gets too small for that to be viable, it will form an oxospore, a different kind of cell that then will combine with another one, the too small cell of another diatom, and they will combine their genetic material and split off, and they will make two new big cells of the big one. So it's not exactly sexual reproduction, but it's a way to mix genetic material. I mean, it's not at all sexual reproduction, because sexual reproduction, as we talk about here, is exchanging one, or exchanging or one half a set of gametes, making a specialized gametes that combine. So pretty cool. That is for binary fusion. Other ways that that reproduction is very bizarre in the alien world is many organisms, especially in the metazoans, they don't bother with having any males at all. They are parthenogenic, meaning the females give birth to fertile young. So rotifers and cladocerans, there are lots of these in which there's never been observation of a male organism. So either they look like something completely different. Cladocerans are like Daphnia, that ostracod that I showed you was a cladoceran. Rotifers, we're gonna see some. Rotifers are I don't like to guarantee what we're gonna see when we look through a microscope or go out in the field, because I've had bad luck doing that. but I am highly optimistic we'll see rotifers there. All of these are metazoans. They're multicellular animals, and rotifers have these two cool circular things of cilia at their heads. If you well, they are at their heads. We'll see some examples. They make feeding currents that then they feed, and then cladocerans are these copepod, or not copepods. They are arthropods. So these little guys, but many of them they just give birth to females, and some of them even the Daphnia, they give live birth, and they give live birth to gravid females. So the females are already pregnant when they are born. If you can imagine that. How strange is that? And so the question is, well, how do they exchange genes? How do they have genetic exchange? And I am unaware of any good explanations for that, for all of them. There have been some where there's some thoughts that there's enough, because these organisms are so small and they're living in the surface water, they get zapped with a lot of sunlight and UV rays, and so it damages their DNA, and they can do self-repair on their DNA. And the thought is, well, they're just pretty crappy about repairing it exactly the same. So their DNA is essentially evolving all the time. Horizontal gene transfer is something where it's usually aided by bacteria, where genes are moved from one organism to another. That's another hypothesis for how this happens. And I don't know what else it might be. So check this out. This is a Daphnia, which looks a lot. See, it looks a lot like that little ostracod. This one's a little bit squished. These are two babies that it is carrying. So they have live birth. And we can watch a little bit of this video. This one, at least I think we can. Oh, that's not looking good. Oh, there we go. All right. So this is a metazoan. It's got kind of everything we have. Here's its heart. Its eye spot. Here's its gut. And it's got its legs down here. Here's mouth parts. And its feet and everything. And it's not moving because the slide is losing water, so it's kind of squished, which meant I could get a really good image of it. And you can see, so it's about 500 microns. That's about half of a millimeter across. So these are what are called water fleas, Daphnia. Oh, and here it is, right side up, if you will. And it's got, yeah, so it's got these little babies in it. So live birth at the micro level. That's a little strange. All right. Some of the other things to think about is that for the physical environment is the size and the stability of where these organisms live. And so they are microns to millimeters, and that means you can find them in things like small puddles, shaded areas. I have some cool things. I don't think I included any today, that are in the cracks in my street outside, just in the pavement on the asphalt. Or and you know how hot that is, but I'd encourage you, go out and look at your street, especially if it hasn't been paved in a while like mine. And if you've got cracks in it, look and see if there isn't some moss growing in there, because dirt gets in there. You get a little bit of moss in there. You can bring a sample of that on Saturday, and we'll put some water on there, and I think you will be surprised at the life that is living in that horrible environment. Because frequently these environments are ephemeral, meaning that they are there at one moment and they're not at others. I'm going to show you some videos in a bit of this amazing bloom of Euglena, these green single-celled organisms that I found, and I've only seen them a few times, but where I see them is alongside the road in a mud puddle that for 365 or 4 days of the year, that mud puddle is just a mud puddle. And one day I'm driving by, it's like, holy cow, it's bright green. And so I'm weird enough, when I see that, I stop. And I usually have a water bottle with me, and took a sample. And I got this amazing bunch of Euglena out of there. And then I tried to go back, and literally, this was while I was going to and from Kaka. I went and the next week they were gone. I looked all over for them. I dug in the yeah. So they come up, they reproduce when the situation, when everything is perfect, and then they go back into these resting spores. And it's not like they're in, or at least for most of them, I don't know what Pfiesteria do. Most of them, it's not like think about like a 17-year locust or something, or a moth that spends most of its life in one form and it just comes to another. These things just are inactive most of the time. They just go into a resting spore. All right. And so why is that good? Because the places they live are so small. They have these very frequent wet-dry cycles, frequent droughts, and they are subject to extremes of heat and cold. And so you better be tough to live there. And some of the fun things we can try on Saturday is we'll be collecting some mosses, and I encourage you to bring some from your house. We'll talk more about what to bring at the end, but we can look at them under the microscope, a dissecting microscope. We probably won't see anything unless it's been raining there and they're wet. But then you put some water on them, and within 2 to 3 minutes, rotifers will just come popping out of nowhere. Apparently, they are just there, dried up, tough as nails, waiting for a few drops of water, and then they're going to go to town, feed, reproduce, and then dry back up. Crazy. All right. So let's see, water, we talked about this. Water is as viscous and dense as Jell-O. Diffusion is about equal to local stirring. So you can't really get anything isn't going to diffuse in. So you need to move to greener pastures to get food. So how do these do it? You can either make your own food with autotrophy, which is like photosynthesis or chemosynthesis. You can get your food from somewhere else by eating someone else with grazing or predation, and you can do both, which this is where that plant versus animal thing really breaks down, is mixotrophs, who can do both photosynthesis and they consume other things. So let's, enough, I've been talking too long now. Let's look at some more pictures and videos. Autotrophs are what we think of as regular green plant, or as plants, and mostly green plants. And so they can be gorgeous like this. This is a desmate cell, a true chlorophyte, a green algae. This is just one cell. The nucleus is actually right here in the middle, but they can be an incredibly pretty patterns like this. This comes from Kaka. All of these things are coming from Kaka. Here's, if you ever see that floating mats of green algae, lots of times it's like this. This is Spirogyra. It's a filamentous green algae. These are the individual little chloroplasts in it. That's what you see up here too. Chloroplasts are the parts of the cell that contain the chlorophyll. And in Spirogyra, they are arranged in spirals. Hence, its genus Spirogyra. This is a desmid, and simpler but beautiful. And they are fun little organisms. Hopefully we'll get to see some because you can really see their cellular material moving around. These guys don't, even though they don't have organisms or organs in them, they have like organelles and they are moving stuff around with their in their cytoplasm. And then these guys have these little strontium crystals. That doesn't seem quite right, but it might be. But they have these little crystals in them that are just vibrating around at the ends of their cells. I'm unaware of anyone knowing why. What was that? Oh, that was a video. Let's see if we can see it. You don't usually think of a plant cell being that active, do you? Well, but they all are actually. Even on our tree leaves and stuff, there is a lot of stuff going on at the micro level that we just never see. All right, and this is it's not a green algae, but it is another algae, a cyanura, and it is oh, come on, there we go. It's a flagellated. See these little flagella coming off. So it's a group of sister cells. So like those diatoms that we looked at earlier, that were gliding next to each other. These, when they divide, they just stay stuck together and they have their flagella out stirring around. So they move as a whole, as a little colony, into new water because they're plants or they're autotrophs. So they need, just like any other, like the plants we think of at the macro level, they need light, they need nutrients, and they need water. Water's easy to get. Light, they can control their buoyancy to some level, so they stay near their surface. But nutrients, if they are in water and they absorb everything near them, they've got to find more nutrients. So they either stir it with their little flagella, or they're just moving around randomly to find a better place to be. Pretty cool. And then diatoms. Here's another pinnate diatom, and you can see it's rich and it's rafy. A rafe is right down the middle. You can just see a pore of it right here, but we're really not focused on it very well. So I don't know why my videos are starting black here, but let's see if this guy starts gliding for us. These other little dots that you can see are just little bacteria. So that's what a bacterium looks like. So they're kind of boring under the microscope. Maybe this guy is too. I don't know. Let's see. Oh, here we go. Now, oh, that was me. Yeah. So it's what we usually think of as a plant moving around. All right. So those were some of the autotrophs, things that make their own food. Here's some heterotrophs, or grazers and predators. And this is a pterotrich. And it is a single-celled organism on a stalk. There's a little colony of them here that we're going to move here once I focus in and out. And you can see they have cilia up here that they are setting up these feeding currents. Yeah, I'm not impressed with Microsoft's compression album. But let's see, I think we'll get a better one here. What's the time? 7:50. I'm pulling the plug on this. Let's go, because here's some other. So creating feeding currents. Let's see what this guy does. So here's another ciliate here, but this guy is usually just goes around and grazes. And then we have a little vorticella there. We're going to look at them in more detail right here. And I think in the next thing, come on. These are one of my favorite organisms. Again, This is on the root of duckweed. So I want you to be looking at duckweed and we're looking at individual cells on the root here. And these little microorganisms are ciliates, and they are setting up feeding currents. You can see his stuff is moving in and out. And these guys are just kind of drifting around. And then occasionally we should see one of them pop down. Or here, oh, I covered it up. Because they have this line that goes down here. It can contract, and it looks like a spring. So that if they get something tries to get them, they can try to get out of the way. Because, microorganisms need to do everything we need to do. They need to feed, they need to reproduce, they need to try to attempt not to be eaten. Yeah, this you can't, maybe with the compression makes it a little bit harder, but can you tell how there's a whole circulation going on here? And this thing actually will be, I think it actually happened at the beginning. It's going to digest, or it's going to ingest a bunch of food. There it is right there. It's made a new food vacuole, and it's ejecting the water that it's collecting through this contractile vacuole that it has. So just like we eat and excrete and get rid of water and food, they're doing the same thing in one cell. It's a single-celled organism. All right, and this is just one of my favorites because it is cool. Come here. This is a rotifer. So this is a metazoan. Most of them that we're going to see will not look like this. This is an armored rotifer that really is a planktonic form. So it's got spines to stay in, stay afloat. And you can see its feeding currents that it's getting. And then these are its mouth parts right in here. And it actually has two jaws that crush stuff up. Here it is excreting. And it can move these spines when it really wants to. Most of the time, it's just cruising along using the whirling of its cilia bands to move. But when it wants to, it can use those spines to move around. And we've got some other different protozoans. These are ciliates of various types. Oh. And then we've got grazing is always good. Another are ostracod. Here it's actually that same one we were looking at before, but with the lighting difference. So you can see how he's using his appendages to manipulate things and just feeding stuff into his mouth parts. Hers, I should say hers, because most of them there are no males. And then, of course, active capture, which I didn't actually capture. This is a gorgeous little green hydra on duckweed. I love me some duckweed. So we're going to be looking for that. And this again is a metazoan. So it's just like, it's like a sea anemone, only it's very small. So that's 1/10 of a micron here. I mean, 1/10 of a millimeter. So this is maybe 1 millimeter long. So you can see them with your naked eye. But even though it has active capture, it has incorporated chloroplasts in it from things that it has eaten. So it can photosynthesize. Pretty cool, huh? And we are getting to the end. Here's the promised euglenoid, euglena. These are what the first person to see stuff with a microscope saw. One of the organisms where these euglena with a little red eye spot and white at the end. They actually have flagella that are coming out of the front here. That's the way most flagellates swim. But they do this crazy thing. They have what's called plasticity look at how they can change their shape and move around. And so here we are at a higher resolution with some different light. Really fascinating. And totally bizarre. Whoop. All right. Well, that was I'm man, what am I doing? I'm going, there we go. Sorry. Dinoflagellates, another, this is a flagellate that can photosynthesize. This turned out horrible, wasn't worth the effort. All right, but that's one of the red tide organisms, or at least like them. And here is another ciliate. Oh, but the dinoflagellates also consume stuff. So it's like a plant that eats. Here is almost like an animal that can photosynthesize. So these are stentors. You can see some of them swimming around. These that are here have cilia and a band right here, and they're setting up these feeding currents and drawing water in, and then ingesting and filtering out organisms from. You can see they are totally covered with cilia. When they want to go to a new place, they just let go at the base where they've got a little, almost like an adhesive disc or a part of them that, oh, there we go. And that same vortex of water drags them off through the jelly-like water that they're in. And so they move off. Really pretty. And I don't know if we're going to see these. Most of these that I'm showing you came from winter samples. So we'll have to see, January 2023. Here's another one, but again, it's related. It's another stentor, but this one is red and pink instead of green. And I'm trying to there we go. You can see the cilia as it starts to feed. So it was withdrawn, and it'll set up a nice little current here and trying to ingest stuff in. It's hard that the cilia are actually beating much faster than we see, but we're getting the effect like a lot of times you do if you're watching a prop plane propeller on TV, right? Where it looks like it moves in one direction, then another direction as it has some aliasing in the imagery. That's what it's called. All right, and then my personal favorite, these are, oh, come on. These are ciliates out of the guts of a termite. So termites, as it turns out, are not actually that good at, they can't digest wood. What they do is they eat wood and put it in their guts, where these microorganisms, look at how crazy this guy is with all these lumps on him. And then these guys I like to call like the headbangers or the rock, what do we call it? Hair rock or hairband rock and roll guys. And moving around. And then these little filaments we're seeing here, those are some of those corkscrew shaped bacteria. Because really most of the digestion of and the breaking down of the cellulose that the termites are eating is done by bacteria, but they are done by bacteria in the guts of this amazing microflora that is in here, and they're all ciliates, and they have these super long cilia. They are famous for that. So we're going to try to find some termites to look at too. and do a little dissection if we can see some of those. Pretty cool, but I'm getting right up to my end. And here's a bonus feature. Here, I haven't shown a tardigrade yet. I know because last week I did an advanced training. We had a tardigrade in a sample that Christina took home, and she got to see her first tardigrade. We have the same moss that came from is out there because I left it there. So this is what, looking under a dissecting scope with a light shining from the side, this is what a tardigrade looks like. And these others are all the rotifers I was talking about. Lots of rotifers. So the tardigrade's got 8 little legs. It's a little slower. Rotifers do this leech-like movement, kind of up and back. They don't necessarily, the color, this is odd that there was such a color contrast in here, but it made for a good video. All right, folks, that is all I have. I would be more than happy to share. I guess I'm still sharing my screen. To answer any questions if people have any.
Dave, your scales on the slides, the underneath the 10 times, that's magnified 10 times, like 100 microns magnified 10 times. Is that what you're showing?
No, it was so the 10x. Let's see if I can get one of those. Let's just. I'm not sharing anymore, right?
No.
Okay, so. All right. So this, yeah, what this is, it says I was using a 20x objective, meaning that I was on 20 power on the bottom part of the microscope. But that only tells me like, okay, that doesn't actually tell me any scale because it depends on the camera and how big you're making. So this, though, tells me that is 100 microns.
Okay. All right. Thanks for clarification.
Yep. Happy to clarify. And here's what those things look like when I took most of my images. That's what those things used to look like. But enough of that. Stop share. Other questions?
Dave, this is Betty.
Hey, Betty.
Hey, this isn't really a question as much as a comment. I find it's so weird that one that had the 2 little babies inside of it.
Yeah.
It's weird that they were just sort of, the babies were just sort of inside of it, but there was no like reproductive, like there was nothing holding those babies. They were just kind of floating around in the innards of that thing, unattached to anything, it looked like. That's so strange.
It is strange, and I don't know enough about their actual reproductive process and that whole pregnancy. Yeah, to know whether there is some uterine equivalent.
Yeah.
There should be something that holds them. And I'm sure there is, and it's probably just so transparent that it doesn't show up to us, I would guess.
And then how do they get out of there?
Yeah, you know what? They just come out through the shell there, I think. I don't know. It's weird. Tardigrades actually, when they molt, they lay their eggs in their old skin. So sometimes you can find tardigrade sheds, and we'll have to see if they have molts in them. Tardigrades are another one. They're parthenogenic. Yeah, so lots of female tardigrades and rotifers and
wow.
Yeah, it's weird. And there have been pictures. We could probably Google it, but of, of a Daphnia with an embryo in it, and that embryo has an embryo in it.
Wow.
So it's like giving birth to your own granddaughter yet.
I mean, the whole topic, the whole presentation just sort of really makes one reconsider or rethink everything around us, like water, like standing water and like everything. You just never think that there's a whole world of reproducing life in all of that. So it's very cool to look at those.
Oh,
Dave.
thank you, Betty, because that is exactly what I was trying to get across in my kind of awkward way and give you some of the reasons for it. Is it's wondrous in the sense of fills me with wonder. It's like it is so bizarre, and it's all around us, and we just usually are unaware. So thank you. Total
Thank
success.
you.
At least one person to recognize that. Yeah.
Thanks, Dave. Any other questions before we sign off for the evening?
All right. Well, hey, let me say, if you're going to come and if you were undecided, I hope I've piqued your interest, so that you will come. If you have an aquarium, one of the best spots to find cool stuff is in the filter, is in your dirty aquarium filter, and that filter media on the back. So take some of that out and do it the morning before you head out, because it's it doesn't want to go anoxic. It'll kill a lot of stuff in there. It's usually, it needs oxygen. But other than that, get a sample of some things that look interesting to you if you're going to be there. And water is usually boring. You want the plants or some of the mud at the bottom mixed in with the water. But plants are where the action was. There's lots of those things I was showing were attached to plants. So.
Very cool. And thanks again, Dave. And you do have some comments there from some people that I think might have left us already.
Okay.
But again, for those of you that might have come in late, Dave and his wife are moving out of town at the end of this month. So this could be our last moment to share with him and say thank you. So if you would unmute and say thanks to Dave.
Thank you, Dave. Been amazing to get to know you.
Yeah, good luck with everything, and then we'll see you
Yeah.
Saturday.
Thank
Anyway.
you, Dave.
You're going to be missed, Dave.
Dave.
You're going to be missed.
Yeah.
Y'all too, but I plan on at least keeping my membership going. So hopefully I can make some think and drinks. Walk and talks will be tougher, but all of my grandkids, I now have 4, and they're all in South Carolina. So I won't be that much of a stranger.
All right.
Wow.
Good to know.
Does
Thanks again.
this transfer to Arizona, Dave, that your membership here?
Oh no, it's a completely different thing. There's a yeah. That's a whole. Did we stop recording?
Yeah.
Okay, good. Yeah, that's a whole different thing.